is Valeria Gonzalez. I've been interning at the Molecular Pharmacology Lab investigating the effect of various anti-diabetic drugs on intracellular signaling. Let's start with a brief overview. What is diabetes and obesity? Obesity is characterized by obesity. Type 2 diabetes develops from impaired insulin secretion and thus insulin dysfunction without controls insulin secretion. Let's dive into GLP-1, a GLP hormone which manages blood glucose levels and leads to the feeling of satiety. Due to its effects, it has high pharmacological potential to counteract diabetes. The peptide sequence illustrates GLP-1, secreted by the L cells in the distal ileum of the small intestine. The issue, or more like the potential target, remains in the fact that the incretin hormone gets rapidly degraded by DPP-4. Next, we have extendin-4. It was discovered to be an agonist of the GLP-1 receptor. Its half-life, however, is extended due to the amino acid changes in the sequence. It makes it less susceptible to degradation by DPP-4. This led to the development of semaglutide, a GLP-1 receptor agonist that has a weak long resistance due to its structure. The fatty acid and the substitution of the iodine in the 8th position decreases the cleavage potential by DPP-4. I'm convinced that you've heard of the conventional name of this drug, Ozempic. Another drug is also gaining popularity, one that targets both the GLP-1 and the GIP receptors. These receptors are key in insulin decrease and satiety, followed by synergic effects or single tirzepatide, a drug on the market known as Mounjaro, which also has a 7-day half-life. This has shown enhanced anti-diabetic effects due to targeting of both receptors. The question that I investigated throughout this internship is then this. The GLP-1, extendin-4, semaglutide, and tirzepatide differ in terms of GPCR dynamics. Before diving in, a brief overview must be given in terms of the intracellular signaling upon binding of the GLP-1 and other agonists to the receptor on the plasma membrane. The GLP-1 receptor is a 7-transmembrane receptor. When its ligand binds, it recruits G-alpha-S. This G-mobile subunit binds to alanine cyclase, leading to the intracellular release of cyclic AMP. The GLP-1 receptor signaling is terminated by either the internalization of the receptor and the binding of beta-arrestin to it. The main objective being the regulation of insulin secretion when high glucose levels are present in the bloodstream. These are the signaling pathways which I'll be focused on for the past 8 weeks. How do we quantify the signal? Well, it is done through bioluminescence resonance energy transfer, a technique known as BREG. The proteins, which are hypothesized to interact, are tagged with a luminescent and a fluorescent tag accordingly. If there's protein-protein interaction, the photons from the luminescent protein will be passed to the fluorescent tag. If there's no interaction, there's no energy transfer. The assay allows to quantify a gain or loss of signal accordingly. The results from such experiments are graphed as follows. The sigmoidal curve in red represents a full agonist, a drug capable of reaching the Emax, which shows the maximum efficacy of the drug. The curve in yellow shows also a full agonist that is less potent. We therefore need a higher concentration to reach the Emax. In blue, we have a partial agonist, where regardless of the dosages, it does not reach the full effect. The EC50 value represents the concentration of the drug needed to have half of the effects. I will now present an overview of what I've learned, but most importantly, the results. To run any assay, the cells must be maintained in ideal conditions. I was introduced to cell culture. Plasmids from transfections must be prepped. This is done by spreading transforming bacteria onto an agar plate and selecting them via antibiotic resistance, followed by a macrophage to obtain the plasmid of interest. Once created, we transfect the cells with them and validate the transfections through RNA isolation, cDNA synthesis, and qPCR. When the transfection is successful, then we proceed to replating into a 96-well plate. The agonists are given and the results collected. These are them. The dose response from G-alpha-S recruitment shows that the endogenous GLP-1 and semaglutide have full agonist potential at lowest dosages. They are therefore the most potent. Tirzepatide shows to be a partial agonist, even at the handiest dose concentrations. On the right, we have the G-alpha-S recruitment temporal data at 1 micromolar. It can be seen that both the GLP-1 and semaglutide showed the most similarities in G-alpha's recruitment, whereas the efficacy of extended 4 is reduced. I'll be showing this table to keep up the results throughout. Regarding cyclic AMP, extending 4 showed to be the full agonist at higher concentrations. Again, GLP-1 and semaglutide follow the same trend, but this time tirzepatide follows as well. The temporal data once again corroborates these findings. This time, there appears to be persistent signaling regardless of the drug throughout time. Regarding the internalization of GLP-1 receptor, all drugs tested, excluding tirzepatide, reached the Emax. This is thus the case for tirzepatide, suggesting that the drug does lead to little to insignificant levels of internalization. The temporal data shows the signaling being increased in time for all drugs, but the amplitude of the response when tirzepatide is administered is massively decreased. Finally, we have beta-arrestin recruitment assay. It is clear that GLP-1 shows full agonist potential at smaller concentrations. Therefore, it has the highest potency, followed by semaglutide and extending 4. Tirzepatide showed no beta-arrestin recruitment. Why are these important? To keep the receptor active, the ligand must keep its signaling potential. Tirzepatide overall showed the least internalization and beta-arrestin recruitment throughout. During my time at Helmholtz, I also undertook some confocal microscopy, which I'm happy to share with you today. It is yet another technique to validate transfection. It can be seen that according to the type to which the GLP-1 receptor is coupled with, there are different outcomes. The GLP-1 receptor coupled with GFP internalizes. Such can be seen on the video in the middle, whereas the receptor coupled to the STAMP tag appears only at the cell membrane. This is because the substrate is impermeable and does not penetrate the cell membrane. Other techniques I have acquired during my time here include islet isolation, including the injections with collagenase, and several brain dissections. Prior to the end, I would like to take some time to thank the scientists who have been guiding me along through the internship. Professor Timo Müller for having me in his lab, Dr. Aaron Novico for the project, Karen Copeland for the patience and introduction to cell culture, and as well as Ezgi for her supervision throughout this time. I would also like to thank Daniela for teaching me most of the in vivo techniques. And last but not least, Melanie, Kalina, and Kathleen, your colleague, for making this experience possible.